Hi guys. So I'm going to kind of review the beginning of the scientific method that we went through yesterday, and then we're going to talk about the Komodo video. So I'm going to use the Komodo video from the beginning of it to just review. So observation from the Komodo video that we saw was that it only takes one bite for the Komodo dragon, the lizard guy, to kill the buffalo. We came up with the question yesterday, okay, well, how does the Komodo kill this big gigantic buffalo with one bite? And now we had a hypothesis, and I just came up with one, and I said, well, if the Komodo kills the buffalo in one bite, then maybe it uses snake venom or some kind of poison it has in its teeth in order to kill the buffalo. All right. The next thing we're going to do is conduct some type of an experiment. Now, if we were in the classroom, how I do this every year is I say, all right, what would be a good experiment to do? And a lot of students say, well, bring a Komodo dragon in. Yeah, that's what we're going to do. We're going to bring a Komodo dragon into the classroom and just let it roam, and it'll be a lovely time, and none of your parents will complain, right? Wrong, right? So we don't want to do that. They're obviously dangerous. They can kill a gigantic buffalo with one bite, and you guys are much smaller than a buffalo. So we don't want to have any contact with a Komodo dragon, all right? But an experiment doesn't always have to be completely hands-on, guys. An experiment could even be, okay, let me do some research. Let me find something about the Komodo dragon, all right? Or even look at a picture. So what I did here, guys, is when I was making this lesson years and years and years ago, we went to the Pittsburgh Zoo. And the Pittsburgh Zoo just had its old Komodo dragon die, which, yeah, that kind of was sad. But then they got two new ones in, and it was like opening weekend, and I got to see the new Komodos, and I was totally geeking out, right? And I was taking pictures because I like to take a lot of pictures, and I got this picture of a Komodo dragon, and it was absolutely perfect. So let's talk about the experiment before I show you the picture. Now, our hypothesis was that maybe there's some type of venom. Well, we'd have to look at the Komodo dragon's teeth for that, right? Because we know venom comes out of like the fang-like teeth, and they have a little bit of a hole in the bottom so the venom can be unleashed. So if our hypothesis is correct, we would hope to see fangs in the lizard's mouth. Here's the picture. Oops. There we go. So looking at this picture here, you can see the teeth, but there's no fangs. It does not have retractable fangs either. All right, but we do see something else, and this is why I love this picture so much. We see slobber just everywhere. Okay, there's slobber kind of on its tongue. There's slobber all around its jaw, and it's even connecting the bottom teeth to the top teeth. So let's watch the rest of the video here.
Soon the buffalo's wounds are festering. Within days, a powerful infection destroys its body, and it dies. It's a puzzle scientists all over the world are trying to solve. Only one thing's for sure. It works. Don't expect leftovers from this gruesome feast. Almost everything is eaten. Bones, hooves, even hide. It's like a scene from the age of dinosaurs, but appearances are deceptive. For a lizard, the Komodo dragon is a relatively new arrival on the planet.
That was crazy, right? So the Komodo actually has bacteria containing spit. All right. So, yeah, I know you like the picture. We're going to go over control groups and experimental groups here in a minute, but let me talk about the Komodo just a little bit more. So with this Komodo dragon, guys, we have the spit, and the spit contains so much bacteria that it actually makes wounds or infections in the buffalo, and the buffalo dies days after. So the Komodo, I believe they can smell. It's like a crazy thing. They can smell miles away. So they know when that buffalo is like starting to, I guess, decay, which is kind of gross to think about. And then the video said that they also eat everything. They eat the bones, the hooves, and the hide. They cannot digest bones, okay? But they eat them. If you guys have ever seen, if you've ever eaten the chicken wing, okay? So you get the meat part in the middle of the chicken wing, and then toward the ends on each side, there's like all this cartilage and tendons and stuff like that you can't quite get the meat off of. Komodo dragon doesn't worry about that. What it does is it'll take those little parts off the middle, just like you guys do, and then it'll take the rest of the chicken wing, throw it down its throat. Its stomach will actually digest the rest of that stuff that we can't eat off of it, and then it'll take those bones and go like kind of like a cat coughing up a fur ball. It'll go and then just throw the bone up, which is awesome. It's kind of a sight to see, right? So that's the Komodo dragon, guys. You can see how we went through the scientific method there a little bit with it. In your hypothesis, mine, I knew it was wrong because I've taught this lesson before, but mine was wrong. And it's okay to be wrong with a hypothesis, okay? We actually learn a lot from wrong hypotheses. We know what not to do and what not to replicate, again, by being wrong. So in an experiment, guys, we're going to have two groups. We're going to have a control group and an experimental group. Now, the experimental group is the reason for doing the experiment. The control group is just to make sure your experiment was set up correctly. Let me give you guys an example, okay? I'm just going to do something very simple here. Let's talk about plants, all right? So let's say you set up, 50 plants, all right? And you have all these plants set up. You put the soil in, you put the plants in, and you want to see what plants can grow faster, okay? What you're going to use is you're going to use only one variable. So the soil will be the same. It'll be the same plant in every single pot. But what you're going to do is you're going to try different, liquids and see if the liquids change the growth rate of the plant. So you know that plants grow well in water. That is your control group. That is what you know. You know for sure that the plant is going to grow well in water. All right. Now your experimental group, we could change a bunch of different things. Okay, let's try, these ones are going to grow in water. All right, maybe these 25 over here. These 25, I'm going to put coffee in, and we're going to see if that does anything, or Pepsi or Coke, whatever, That's your experimental group, and you want to see if the Pepsi, the Coke, the coffee, whatever, is going to make the plants grow any faster. So the experimental group is saying that, hey, something that we don't know about might be actually better than just regular water, but I know the water should make the plants grow. Now, here's another good reason why you need a control group. If the water doesn't make the plants grow, then, oh no, something's wrong with your experiment. Maybe the soil's no good. Maybe the plant seeds that you got were no good, okay? So it could be a bunch of different things. So the control group is nice because then you can see if something went wrong with your experiment. We're also going to have independent and dependent variables. We're going to go over some examples. I have some written up at the end. But here's what an easy way to remember independent and dependent variables. It's cause and effect, okay? The independent variable, that's going to be your cause. The dependent variable is going to be your effect based on that cause. When I was student teaching, guys, a long time ago, and you don't have a lot of control when you're student teaching, the teacher that I was shadowing, he did this experiment with goldfish, and I don't like doing a whole lot with real live animals because I don't like to hurt animals, but what we did was we took these goldfish, and every student lab group got a goldfish, and we took them out of the tank, put them in water, and then what we did was we slowly adjusted the temperature of that water. And what we were seeing is if the breathing rate of the goldfish slowed down or sped up with the increase or decrease of the temperature of water. So independent variable, guys, that's the cause. Again, as I said, it's what you have control over. So in that experiment, guys, we took the goldfish out, we put them in water, the students changed the temperature of that water, and then the breathing rate of the fish changed, okay? What did the students have control over in that example? In other words, what was the independent variable? Well, it can't be the breathing rate of the fish unless you're going like, breathe, fish, breathe. And then the fish is actually listening to you and it's like, okay, I'll breathe faster, I'll breathe. No, it doesn't work like that, okay? What we had control over, what the independent variable was the temperature of the water. We were able to change the temperature of the water. And then based on our change in the temperature, the dependent variable was then the breathing rate of the fish. So that's the effect. You changing the temperature, independent variable, okay, caused the fish to breathe faster or slower. That's our dependent, okay? So there's a good example there for you guys of independent and dependent. We'll do more of that later. There's different types of data. It's very easy to show what the data is, if it's quantitative or qualitative. Look at quantitative, guys. We got this little word in here called quantity. Quantity is always like number. If you ever went to Walmart, Giant Eagle, wherever, and you had to like get bananas or something in the produce section, you go up to like the self-checkout area and it'll say enter quantity, and you'll say like, oh, I have 2 bananas, I'm going to enter the number 2, you know. But quantitative data, it's quantity. It always has a number. If you see a number, it's quantitative, okay? If it does not have a number, it's qualitative. And that's that guy down there, qualitative data. So if you wanted to say like, I don't know, I weigh 150 pounds, that's quantitative data, okay? If you wanted to say that, I don't know, Albert Gallatin School District is 15 miles from my house, that is quantitative data. If I wanted to say, hey, Susie's wearing a red shirt today, that's qualitative. There's no number in it. Or hey, you know, that ground is hard. That is qualitative, okay? There's no numbers assigned to anything. That's the difference between quantitative and qualitative data. So done with the experiment section, guys. Discussion, results, this is where you talk about all your findings, all your different things that you had. It shows the outcome of your hypothesis, all right? So, and I said this before, even when a hypothesis is not correct, it's still good because other scientists can use your data and be like, hey, you know, they already tried this, it didn't work. We can't do that, or we shouldn't do that again because we know it's not going to work. But anyway, this is where you talk about all of your findings, okay? You say, hey, this data meant this data meant this, you know, all different things like that. So the conclusion is your last part. This is where you kind of analyze everything you said in your results, all this data that you just kind of went over. You want to say, all right, what's the outcome of this? What can I take away from the experiment? What's my overall message I want everyone to know based on everything I did? And that's your conclusion, guys. And it's just like a conclusion in English, okay? It just sums everything up, discusses all your outcomes, and that's it. So this, I'm going to show you one more video for the day, guys, and then, well, 2 more videos, and then we're going to be done, all right? So just to go over the scientific method one more time, this video does not have sound, just because it was taken in the deep ocean. So here's a shark. This guy down here is a hagfish. They put some hagfish bait out there to try to attract the hagfish. And this is all the way on the ocean floor. That's why there's no sound. Shark bites the hagfish. Shark swims away. Hagfish lives. Another fish. You guys can see the hagfish there in the middle. Fish bites the hagfish. Fish swims away. Hagfish lives. Another guy. Oh, he engulfed the whole hagfish. Oh, spit him back out. And the fish swims away. Okay, so I don't want you guys to see the rest of it because it tells the answer of everything. But this is our hagfish. You guys saw what happened with the hagfish, okay? Now you might be wondering, okay, why does it live on the ocean floor? That might be a good question, But what's going on in the video? What's the plot of the video? Well, our observation should be that, the hagfish is being swallowed by these different fish, and the shark is being spit back out. It's not being killed. And why are the fish not eating it, right? So, that's your observation, guys. Your big observation is that the animals or the fish are not able to eat the hagfish, okay? Question is, why? Why are they not able to eat the hagfish? Well, you might have a bunch of different hypotheses because the hagfish kind of looks like, an electric eel. So a good hypothesis would be, well, if the fish spits out the hagfish, or we could say the shark spits out the hagfish, if we want to be a little more specific, then the hagfish might have electrocuted the shark, okay? And that's a good hypothesis, okay? Because it looks like an electric eel, all right? So this one's a little bit more difficult to do an experiment with. So what I did was I found you guys a video on it, and I will show you what happens with the hagfish here. Video's a little blurry, guys, so I apologize.
Now it's recording. We have 180 seconds to record.
Okay, yeah.
So they actually brought a hagfish up, and that's a hagfish chilling there.
So I'm collecting some slime.
Yeah.
Can you see?
Oh, yeah.
Gonna mix it. And my official mixing apparatus. Let me hold it up.
You gotta
so
back
everybody
off a
can
bit.
see.
Wait a minute. Well, okay, go for it.
And the entire beaker.
Wait a minute. It is empty. Turn the beaker upside down and pour it out. Nothing.
Okay, so there's your video on the hagfish, guys. If you want to see what the inside of the hagfish's mouth looks like, oh my goodness, it's actually like a jawless fish. It looks like something out of a Stephen King horror movie, but I will take that off because that is horrifying. The experiment they did, we don't have the money to do that kind of thing. That would cost, to go get a hagfish down at the ocean floor, would cost a lot of money. So they shocked the skin. They provided a stimulus for the hagfish, and its response was that it actually turned the water around it into a slime. The hagfish is fine. It's able to breathe in that. But what happened was all the other fish that were around it, they got their gills clogged up with this slime, and eventually they probably died if they ingested enough of this slime. So that is one of the coolest defense mechanisms I've ever seen out of an animal. So that's the cool thing about the hagfish, and a nice way to go through the scientific method and seeing how the hagfish works. Let's get rid of that one. Okay. Just some examples, guys, and then we will call it a day. A scientist develops a new peanut for people who are allergic to nuts. He gets 60 people for the study. 30 people are given the new peanut, which is group 1, and 30 people are given a regular peanut, which is group 2. After his test, 0 people in group 1 show an allergic reaction, and 30 people in group 2 show an allergic reaction. So what is the independent and dependent variable in this, okay? So independent variable is, what do you have control over? What is the cause, all right? So in this example, our independent variable would be the type of nut given to the people. So the two different groups, whether they are given a new peanut or a regular peanut. That's our independent variable, all right? It's not specific, guys. You don't want to say the independent variable is the new peanut or the regular peanut. It's whatever nut they're given. Okay, that's the difference. And in the dependent variable, what's the effect of giving people these peanuts? Well, it's if they have a allergic reaction or not. Okay. Second question down there, what is the control and experimental groups? Well, the control group, this is the one we know the outcome of. Do we know if the new peanut is going to give people an allergic reaction? We don't yet. Okay, that's our reason for doing the experiment. So our control group is going to be giving the allergic people a regular peanut. We know they're going to have an allergic reaction. We know they are going to blow up like a balloon. Okay. The experimental group, though, is going to be our other group, the new peanut. We don't know if they're going to have an allergic reaction or not. Okay. So first example, guys, again, independent variable, what peanut they're given. Dependent variable, if they have an allergic reaction. Control group, that's going to be our group given the regular peanuts because we know they're going to have an allergic reaction. And then experimental group is our group given the new peanut because we don't know yet if they're going to have an allergic reaction or not. Second one, a scientist makes a vitamin that makes people stronger. One group, group 1, is given the vitamin, and one group, group 2, is given a vitamin that simply has orange juice in it. After 2 weeks, they test to see if the people are stronger. What's the independent and dependent variable? Well, for this one, guys, our independent variable is what pill they are given. That's what the scientist has control over. Our dependent variable is going to be if they get stronger. Okay, so independent variable, what pill they're given. Dependent variable, that's going to be our effect. It's if they get stronger. What's our control group? Well, we know that orange juice should not make people stronger. So the orange juice should be our control group. And then the experimental group is going to be the actual vitamin, the one that the people are given in group 1. All right, guys. So this is the end of today. You can work on your assignment for variables, and I'll see you tomorrow.